Hello and welcome. Thank you for joining us today. My name is Corinth Auld, and I'm the Medical Science Liaison for Mercodia. I'm also joined by one of our R&D department's product development specialist, Maria Schwanbeck. So it is a pleasure to introduce our speaker for today, Dr. Juul Holst. He is a professor of medical physiology in the Department of Biomedical Sciences at the University of Copenhagen, and he is the scientific director of the Novo Nordisk Foundation Center for Basic Metabolic Research. In addition to being a member of a number of editorial boards, he is an elected member of the Royal Society of Science and Letters and has received numerous awards. He is a world-renowned expert in the diabetes and obesity fields with accomplishments that include the discovery and characterization of GLP-1. We are very excited that he has joined us today to talk about Glucagon and Glicentin. So without further ado, Dr. Juul Holst.
So thank you very much, Corinne, and hello to everybody who wants to join this session. It's a pleasure to have you here, and I'm very pleased to be asked to discuss Glucagon assay, as I called my presentation, with you. So the problem, of course, is to measure glucagon, and I've called it glucagon because I mean to talk about not only the pancreatic hormone glucagon, but also other glucagon-containing moieties. So that's why I call it Glucagon assay. But let's see now what happens here. So what I'm really talking about is Proglucagon-derived peptides. And here we have that there's a single gene which is expressed in the pancreas and in the brainstem and in the gut. There's a special thing about birds and fish that they may have splice variants of the precursor, of the RNA precursor, which means that the parts of the precursor will not be processed further in certain organs. But that's special for birds and fish, and we won't talk more about that right now. What is interesting, of course, is that there is a differential post-translation processing so that things come out differently, even if the proglucagon molecule is the same in all these locations. And that is because we have a differential expression of processing enzymes with the prohormone convertase 2 in the pancreas and the prohormone convertase 1,3 in the gut and in the brainstem, and that causes differential processing of the precursor. So here we have an important slide because this shows that glucagon and the products of Proglucagon are members of a large superfamily of related peptides. And this shows you the long list of all these molecules out here with GIP and GLP-1 and exendins and secretin and VIP and so forth. But the important thing here for our purpose is that the relationship, the family pattern here is that of amino acid homology. And you can see that there's a very pronounced homology of amino acids in the N-terminus. And this is important because when we get to the measurement, all these peptides that are listed here are those that should be looked out for cross-reaction in any of the assays that we are going to look at. So that's important. Now let's move on to the actual processing pattern, and this is what we have learned over the years that happens. So up here we have the structure of the first product, the peptide Proglucagon. And it contains, as you can see, of course, glucagon, which is located here in this region. And in the pancreas, glucagon is cleaved out of the precursor, of course. Interestingly, that leaves these two big fragments here. So one is called Glicentin-related pancreatic polypeptide, this one, and the other one we usually call major Proglucagon fragment. Now, if we look more closely at the major Proglucagon fragment, we can see that it contains two units here that are both glucagon-like in terms of amino acid homologies. And actually, if we go back here, you can have a look at GLP-1 and its homology to glucagon very clearly, and GLP-2 also with a considerable sequence homology to GLP-1. So that's why they're called glucagon-like peptides. And there's a 1 and there's a 2, and there's an intervening peptide here, as you can see. And now the interesting thing is that when we have the processing enzyme PC2 in the pancreas, we get a cleavage here and a cleavage here, and that means that glucagon is liberated, whereas this one remains as one piece and is secreted as this piece. And this one remains as one piece and is secreted as that. That has been looked at in detail. And in the gut, we have a completely different pattern where the prohormone convertase 1,3 cleaves proglucagon so that GLP-1 here is cleaved out with an intervening peptide, and GLP-2 is cleaved out, whereas the glucagon sequence remains buried in this big peptide, which is called glicentin. But of course, it contains the entire glucagon sequence. It's a peptide of 69 amino acids, which is secreted in parallel with GLP-1 and GLP-2. Now, part of glicentin may be broken up at the same site as in the pancreas to release GRPP, the same peptide up here. There's not much to say about this peptide. It's probably inactive from what we know. It has been looked at in some detail, but this has really never been published. But it doesn't seem to be extremely interesting. But this other molecule is very interesting indeed. As you can see, it contains the entire glucagon sequence, plus and a little addition here, little octapeptide here. And that, of course, makes it related to this molecule because they have the same C-terminus, but it also makes it related to glucagon because they have the same N-terminus. And interestingly, it is also related to GLP-1 because it also can interact with the GLP-1 receptor. So this is a very, very interesting peptide, and we'll talk a little bit more about that later. Now let's move on and have a look at glucagon physiology. This is just to remind everybody. So what I'm really trying to do here is to answer the question, is glucagon worthwhile? Is it interesting to look at glucagon at all? And so I've just summarized for you a few things here, that it's a 29 amino acid polypeptide. It's secreted from the alpha cells of the pancreas, increases hepatic glucose production, stimulates amino acid metabolism. I think this is very important. I'll come back to that. Increases insulin secretion. It's a very powerful insulin secretagogue. It increases lipolysis in some rodents and in birds, but not in humans. So this is a very important distinction to make, that even if you can find a lot of literature about the importance of glucagon for lipolysis and rats, this does not appear in humans. It also has effects on gastrointestinal motility. It's a complex mechanism. It may stimulate thermogenesis. Interesting. It may stimulate ketogenesis, and does so in the liver. It has positive inotropic and chronotropic effects on the heart. And it's actually been used, as a young doctor, I used to use it in the emergency ward. So it certainly has those activities. Here is a picture of glucagon and its interaction with the most important site in people, in humans, that is the liver and the hepatocyte. And here we have the glucagon receptor, the 17M receptor, that is coupled to a G-alpha or G-Q, G protein. And that means that we can, via the G-alpha, we can activate adenylate cyclase, have a cyclic AMP production, and protein kinase A built up here, which then can phosphorylate and work on, for instance, this kinase that can then activate the phosphorylase, and then we can have the glycogenolysis. That is one of the main actions of glucagon. It can also, pKa, can also phosphorylate and activate important enzymes necessary for gluconeogenesis. So here we have another very important mechanism of action. At the same time, glycogen formation will be inhibited, and glycolysis will also be inhibited. Via this pathway, it can also influence the calcium levels in the hepatocyte, but it is mainly known for its actions on cyclic AMP. It's one of the classical stimuli for cyclic AMP production. So this is an experiment published by Sloop et al. In 2004, where they used a glucagon receptor antisense technology and looked at what happened for glucose metabolism enzymes in the liver of diabetic and non-diabetic rats. Let's just have a look at the Sprague-Dawley rats here, where you can see that this is the gene expression of PPAR gamma after this glucagon receptor antisense. And you can see it's not influenced, so this is just a control gene. You can look at this one, which is glucokinase, and it's also not influenced by the glucagon. But something that is glycogen phosphorylase, which is clearly low or is higher in the activated state. And here you have fructose 1,6-biphosphatase, which is, again, very strongly affected by the antisense treatment. And you have the enzyme PepsiK here, which is also inhibited, and you have glucose 6-phosphatase, which is also strongly inhibited, and finally expression, of course, of the glucagon receptor. So this is really a catalog of what glucagon does in the liver, demonstrated very elegantly by using the glucagon receptor antisense in the liver. So this is really what glucagon does. So how does this pan out in life, in real life? And that has really been something that people have been discussed a lot until it was finally, using various techniques, clear what happened. So here you have an experiment in a rabbit. It's a healthy, normal rabbit. It's conscious. And what is done here is that glucose is clamped. It's maintained at the fasting level. And here, these animals are injected with a huge amount of a very high potent, high affinity glucagon monoclonal antibody. And what that means is that within seconds, all glucagon in the body has been bound to the antibody and is gone. So what happens to the hepatic glucose metabolism? And this is what you can show here, because here it is necessary to infuse glucose to maintain this fixed level of glucose in the plasma. And here you have the infusion rate, and you can see that this is a very high infusion rate. So we're up to 4 milligrams per kilogram per minute. In people, our hepatic glucose turnover is about 2 milligrams per kilogram per minute. Now, this is a small animal, but it's still a very high rate of infusion. So what this shows us is that glucagon is necessary to keep up the hepatic glucose metabolism at all times. Very essential hormone for the maintenance of glucose production in the liver. This has been done with another technique here in people. So what has been done here is that somatostatin has been infused. Somatostatin will inhibit very strongly glucagon levels, as you can see here. Now they're inhibited. But it will also inhibit insulin secretion. So that has to be replaced. So here insulin has been replaced very nicely. There's no change in insulin. There's only a drop in glucagon. So now glucagon is lost again. And again, this is a clamp study. And here we have the glucose levels. They're very nicely clamped here at 100 milligrams per deciliter. But to be able to maintain this, in spite of the fact that hepatic glucose production goes down like this, you have to infuse all of this glucose. And again, you can see here that this is been up to something like 1 to 2 milligrams per kilogram per minute. So again, this tells you that all, with a different technique though, that glucagon is extremely essential for the maintenance of hepatic glucose production. So this is why we need to have glucagon at all times. Now, how does this happen? So the glucagon is released, of course, from the pancreatic islets. And here we have the traditional picture that we all have learned with a mantle of glucagon cells and a core of beta cells that produce insulin here. That's the classical picture. This is just from a textbook. Whether it really looks like that in people is a bit of a question. Here's another concept, which I find much more interesting, where 3D depictions of the pancreas have been made in human islets. And as you can see, this, according to this concept, the beta cells are really wrapped into a mantle of alpha cells, yes, but that means that the vessels reach the alpha cells first and the beta cells next, rather than the classical opinion that blood reaches the beta cells first and then the alpha cells. So I think this has great functional implications. Enough of that, let's move on to see what happens. One of the toys we have been using in my laboratory for a long time is the pancreas, and here we see a perfused porcine pancreas. This is the pig pancreas taken out, so that we can look at the hormonal secretion from this preparation. And in essence, what I want to show here is the secretion of insulin and the secretion of glucagon as a function of the perfusate, the perfusing medium glucose concentration. And you can see that if we are around the, normal fasting glucose concentrations, there's little secretion of insulin and there's little secretion of glucagon. And if glucose rises, then there's a lot of insulin secreted, and if it falls, then insulin secretion is shut off, and there's a lot of secretion of glucagon. This is really the most important lesson we can learn from studies of this. Let us just for a while consider, since this is all about measurement of glucagon, let's consider for a while what happens if there's too much glucagon, if the plasma glucagon concentration is far too high. Let's have a look at that. That's what may happen. Isn't it surprising? Here we are looking at a very peculiar skin syndrome that we call a migrating necrolytic erythema. And you can see that it consists of rather circular lesions, and they start out like a burn, like almost completely like a burn, and then they heal up with some pigmentation, as you can see, and they may become confluent and therefore create all these strange patterns. On the next slide, you will see a similar pattern here. This is even more resembling of a burn, and here you can see that the burst of the layer here, and you can see the healing with pigmentation. That is what we call the glucagonoma syndrome. And to understand this, we could have a look at some of the actions of glucagon here. Let's see. So what is it that glucagon does? Well, as we just discussed, glucagon promotes glycogenolysis and glucose production, and that is by breaking down glycogen, by activating the phosphorylase, creating glucose 6-phosphate, and then glucose 6-phosphatase removes the phosphate and glucose is exported here. But as I said, the other main mechanism was really to promote gluconeogenesis and ureagenesis in the liver. And that occurs with amino acids as substrates. And they then get into this circle and generate glucose and urea. And that means that so much amino acid can really be used for this urea production that the plasma levels of amino acid will fall very dramatically. And that means that the cells in the stratum germinativum in the skin do not get a sufficient supply of amino acids and die. And that's why you get the necrolysis, the lesion in the skin that turns out the way I showed you. So that's what happens when you have too much glucagon. You don't necessarily become a diabetic because glucagon at the same time will stimulate insulin secretion, and then glucose will be kept down. Most people, though, do have some degree of diabetes. What happens if there is too little glucagon could be the next question. And that is not so clear, but let's have a look at it. So again, this is experiments that we did in rats, where we used the same technology of immunoneutralization with large amounts of a high-affinity glucagon antibody to take away immediately, instantaneously, glucagon. So these are postprandial rats. These are the control ones. Here you see that you can lower in these postprandial rats glucose by a couple of millimoles per liter. Nothing dramatic, but very significant. But in 10-hour fasted rat, there was no effect. In 48-hour fasted rats, there were no effects. And that's a nasty thing to do to rats, to let them fast for 48 hours, I can assure you. So this was very surprising. If we move on here and see what happens on 10-hour fasted rats, here we have them, no effect of the monoclonal antibody. And then add propranolol, a beta blocker, or phentolamine, an alpha blocker. And here the combination. Well, you can get some lowering of blood glucose, but nothing dramatic again, mostly because of the effects of these beta and alpha blockers, of course. Nothing dramatic. So the main message here is that glucagon really is not essential for maintenance of glycemia in the fasting state. Cortisol is the important hormone for that, because that provides the liver with gluconeogenic precursors. So lack of glucagon, therefore, does not create severe hypoglycemia unless there is insulin around at the same time. Then it's dangerous. So what really happens if you take away the glucagon part is exactly what we would expect from the original studies we did with immunoneutralization. And here we have post-absorptive rabbits, that you have a lowering, some lowering of glucose, yes, but not dramatic at all. And interestingly, insulin levels will follow the glucose. So if glucose falls, then you have insulin falling. But if you maintain glucose in a clamp, insulin does not fall. So it doesn't really seem to be crucial for insulin secretion in that situation. Here we have plasma major proglucagon fragment. So what we're doing here is measuring the alpha cell secretion. Normally we would measure glucagon, but in this case we have injected the animals with an antibody, so we cannot measure glucagon. But we can measure the major glucagon fragment instead. And you see that it rises dramatically, but if you clamp the glucose, it doesn't really change. So again, we would interpret this to indicate that alpha and beta cell secretion will follow plasma glucose in this situation. The glucagon receptor knockout mice came along and can be used to study the same phenomenon. So here first we look at binding of glucagon to liver membranes. No binding in the knockouts as expected. Cyclic AMP production in the liver membranes. No effect in the knockout mice as expected. And then again, glucose levels. So what do they look like in an animal that doesn't have the GLP-1 receptor? And you can see they fall a little bit compared to the controls up here, but nothing dramatic. Again, proving the point that glucagon is not essential for the maintenance of fasting glucose level. So here is at average levels, and they are a little bit lower, as you can see, yes, but nothing dramatic. This is a challenge, a little bit lower, nothing dramatic. This is an insulin tolerance test, and even that goes well. It's just a little bit lower in the knockout animals. But they do have, as I already alluded to, higher cortisol or corticosterone levels. They are here in this animal. What is the role of glucagon in diabetes mellitus? Well, this is something that has been dealt with extensively by Roger Unger, and he and Lelio Orci formulated the hypothesis that glucagon is essential for the pathogenesis of diabetes mellitus. The presence of glucagon is important, if not essential, for the development of endogenous hyperglycemia and ketoacidosis. And that theory has gained a lot of support more recently, looking at the glucagon receptor knockout mice, where this could be tested in a very robust way. I won't discuss that because there's a lot of things you can discuss here, but let me show you some of the very early data by Jack Jerick here, where he was able to prevent human diabetic ketoacidosis by somatostatin, and this is an infusion of somatostatin in type 1 diabetics, and these results could be interpreted to indicate an essential role for glucagon. Have a look at what happens. So here people are infused with insulin, keeping their glucose levels down, and these are type 1 diabetics without an insulin production of their own. So what happens is that if you stop the insulin infusion here, you will experience a rise in glucose, dramatic rise. These are type 1s without insulin. They also have ketoacidosis, as you can see from the production of the ketone body here, beta-hydroxybutyrate. And in parallel with all this, there's a rise in glucagon. Now, how can we get rid of that glucagon? We can do that with somatostatin. So here somatostatin was infused. Glucagon comes down nicely. And what happens then? Glucose is kept down, and ketoacidosis is kept down. So this was really very important observations indicating an essential role for glucagon in all this. This was type 1. What about type 2? So it has been known for quite a while now that there is hyperglycaemia throughout the day in both obese and non-obese patients with type 2 diabetes. And another interesting observation was made by Bo Arén in Sweden, where he found that glucose intolerance is predicted by low insulin secretion, of course, and high glucagon secretion. He did a prospective study in post-menopausal Caucasian women in Sweden and found that one of the earliest signs of glucose intolerance was an increased glucagon secretion. It's not yet clear how these things develop, but let me show you a few more data. So here's some studies by a Dane, an epidemiologist, Christine Ferk. She was looking in a large group of subjects for incipient impaired fasting glucose and incipient impaired glucose tolerance. Again, over a 5-year period and thousands of people, she was looking at this and looked at who developed IFG and who developed IGT. And she found that these with IFG had reduced hepatic insulin sensitivity, stationary beta cell dysfunction, altered incretin hormone secretion, and inappropriately elevated glucagon secretion. Again, an extremely early sign. Incipient impaired glucose tolerance was characterized by reduced peripheral insulin sensitivity, near normal hepatic insulin sensitivity, progressive loss of beta cell function, and inappropriately elevated glucagon secretion. So in all cases, a very early inappropriate elevation of glucagon secretion was observed. Here are some experiments that we did to try to get to understand this a little bit better. And what we did was to give completely healthy individuals some prednisolone. So here we're looking at plasma glucose, and you can see that you can actually you can make them glucose intolerant, somewhat intolerant with prednisolone. And this was associated with an increase in insulin secretion, and that is because they adjust their insulin secretion according to their insulin sensitivity. So now we just need to have a look at glucagon, and it comes here. Here we have it. See? These are the controls. So this is the control situation before the prednisolone, and then after you get this, which is really completely reminiscent of what you observe in type 2 diabetes. So as soon as you touch glucose tolerance, the least you'll have this diabetic pattern and hypersecretion of glucagon. So this is what it looks like. This is a study we did in 55 patients with type 2 diabetes. And I just want to show you this for two important reasons. So first of all, it very nicely shows that there's a fasting hyperglycaemia in type 2 diabetes. And this was a mixed meal. And it shows you that there's also a postprandial hypersecretion of glucagon after a mixed meal in these individuals. So these are the carefully matched controls. Please also note there's a general misunderstanding out in the literature that the postprandial glucagon response should be a suppression. It is not if the meal is a mixed meal. Only if the meal is predominantly carbohydrates should you expect a decrease in glucagon concentration. Otherwise, in almost all other meals, you will have an increase in glucagon secretion. Now, I love this slide from Phil Felish and his co-workers. It's an old one again. And what does it show? It shows splanchnic glucose production. And over the years, there's been a lot of discussion about what splanchnic or hepatic glucose production really is in type 2 diabetes. Is it elevated? How much is it elevated? And people have really been discussing this because it's always a little bit tricky to interpret tracer data. But this was done with hepatic vein catheterization. So these are direct measurements of the hepatic glucose production. And you can see that there's a hyperproduction of glucose in the fasting state and a much larger increase in glucose output from the liver, or splanchnic glucose output in the diabetics. So I think this is really a very interesting slide. This has been studied in very elegant way in details by Bob Risser from the Mayo Clinic. And what they did here was to study the effect of diabetic hyperglycaemia in man. And so what they did was to look at insulin levels in type 2 diabetics. Here we have the type 2 diabetics and in healthy people. And they did that under two situations, namely one where they had normal glucagon levels and one where they had suppressed glucagon levels. So this was all done after infusions of somatostatin, which would block both glucagon and insulin, and then they were mimicking the insulin patterns of the diabetics or the healthy controls. Here the controls and here the diabetics by infusion. So these are the two situations, suppressed glucagon or non-suppressed glucagon. Here's the insulin, the normal insulin pattern. Here's the diabetic insulin pattern, the delayed and sluggish insulin response, as we all know. So this was one of the conditions. These are the insulin levels. Let's have a look now at the glucagon levels. So remember, the groups were divided in those that had suppressed glucagon. This was done with adjusting the levels so that they had lower glucagon levels. And then those that did not have suppressed levels, they were the kind of diabetic levels. And this was done in the non-diabetic group and the diabetic group down here, those where insulin secretion was adjusted to resemble diabetes patients or healthy controls up here. So what happened then? Well, here we have the glucose levels. So you can see in those where we had normal insulin levels, but suppressed glucagon levels, we have the normal glucose levels. But if you do not suppress glucagon, yes, you have hyperglycaemia as expected. And if you, at the same time, have an insulin secretion pattern of the diabetic way, then you will see that you have a dramatic overshoot here of glucose levels. And this, of course, is very, very elegant proof of the importance of glucagon secretion or the hyperglycaemia in diabetes. And this is also, like shown here, on the glucose production by the liver. Using our technique again of glucagon neutralization with antibodies, we could also do exactly the same, in this case in alloxan diabetic rabbits that we produced here. You can see they start out with high levels, and then we neutralize their glucagon, and the glucose levels can be brought all the way down to normal levels. So this really tells us, these experiments and all those in people, they really tell us that glucagon is essential for diabetic pathogenesis, both type 1 and type 2. So when we get the question, why measure glucagon? Now we can say, because we've analyzed it, that glucagon is essential for both fasting and postprandial glucose homeostasis. It is responsible for a major part of the hypoglycemia in both type 2 diabetics and perhaps also in type 1s. There's some research ongoing now following up on Roger Unger's latest observations, and let's see how that ends. So clinically, when should we measure glucagon? And of course, this is a question of whether we're interested in glucose homeostasis. Where do we have disturbances that are known? We have them in liver disease. And this is not too well explored, so there's a lot of work to be done out there to try to understand the role of glucagon secretion in the glucose intolerance of liver disease. We have major problems in kidney diseases, where there are great abnormalities in glucagon secretion, and this is something that is also being researched in more detail these days, and there's a lot of work to do. I've put here, in general, gastrointestinal diseases, and I'm really mainly pointing out to a number of conditions where we have surprising findings. For instance, in people, any individual with an accelerated gastric emptying, it seems that you will have hyperglycaemia. And that, of course, raises questions whether as to the mechanism of this. Is it related to the gastrointestinal passage or transport, and how is that possible? And this is something that needs to be investigated. This is also typical of patients with gastric bypass operations. They have a characteristic hyperglycaemia, which is also completely ununderstood. And as I've written here, the pathophysiological consequences of these disturbances are really unclear. So there's a good reason to measure glucagon in all of these conditions. And here, finally, it turns out in these days that glucagon may also play a very significant role in food intake regulation. And this is a completely new field, and a lot of work has to be done here. So there are a number of reasons why you should measure glucagon, but the most important is whenever you think of disturbances of glucose metabolism, you should measure glucagon. And I might add, of course, although this has not been done to any extent, that disturbances of amino acid metabolism are also relevant. So, the plasma levels of glucagon, what do they look like? Well, they're determined, first of all, by the rate of secretion, of course. Then they're determined also by the rate of elimination. So where do we have elimination of glucagon? Again, this has not been not very well investigated, but a major organ for glucagon removal is the kidneys. That's where you have the most important elimination of glucagon. What about the liver? There is a tendency in the literature to describe the liver as causing a clearance of glucagon, and that is completely false. There is no clearance of glucagon in the human liver. That is a fact that can be studied very nicely by looking at porto-peripheral or porto-hepatic vein gradients, and there is no gradient. If you look at portal levels and compare them to peripheral levels, you will find that the portal levels are some 2 to 3-fold higher. But that is because the portal blood is diluted into the systemic blood, and the proportion of dilution is 2 to 3-fold, and that's why we have a gradient between the portal plasma level and the peripheral plasma level, not because the liver is degrading or taking up glucagon. The next factor, which is important, is rate of degradation post-sampling. And here there's a lot of rumor going on that there are enzymes that can degrade glucagon. We've just completed a study where we're looking again at the possibility that proteolytic enzymes destroy glucagon after sampling, and doesn't seem to be terribly important. So again, now these are measured plasma levels. What is the stability when the samples are stored in the freezer? Again, this is a factor that must be known or considered. It doesn't seem to be terribly important for glucagon. It seems to survive pretty well, but you can always consider this. So recovery upon sample pretreatment. And what do you do to your samples before you measure them? Do you extract them? Do you concentrate them? What do you do to them? So what is the recovery upon that sample pretreatment? And then if your assay is not reliable, of course you can get any plasma level. So I think these are some very important things to consider when you look at measurements of glucagon. So again, what do we have? We have generally low concentrations of glucagon in the low picomolar range. That means that the assay has to be very good. Not all assays are very good. Typically, we've been using radioimmunoassays. The radioimmunoassay for glucagon was the second radioimmunoassay to be developed at all. And they're easy to generate. And it's possible to obtain a high sensitivity. But of course, they're not very popular any longer because of the use of radioactivity and a lot of technical questions that people don't like anymore. So ELISAs are getting more and more popular, and that is okay if they are good. And as we'll come back to, a sandwich ELISA is really what you should prefer, but that's news. Glucagon assays have been incorporated into various multiplexing preparations, and that is dangerous. I haven't really come across a single multiplex assay for glucagon that works at all. Very recently, people are looking at mass spectrometry, liquid chromatography mass spectrometry, to look for levels of glucagon, and they are usually not sufficiently sensitive. So that's not for general use, certainly. And to obtain the necessary sensitivity, usually there is some sample pre-treatment that is necessary, and the recovery of these processes is something that is very, very critical and has not been looked at in any detail so far. So what you really need to do is to look at the reliability, and I will put up all the criteria in a minute. Here I've just put that the most important test of accuracy is, of course, to do recovery experiments, to do dilution experiments, and mixing experiments, and then also to compare with physicochemical methods, just like the liquid chromatography mass spectrometry that I talked about. That is almost never done by investigators. In fact, anybody who measures glucagon should study the reliability of the assay in his own hands. I think this is very, very important. Most people put some poor PhD students or graduate students to do the glucagon assays, and everything is all over the place, and you cannot trust any of the values. Some people write that they have been using a validated assay, and that also always annoys me a bit because, validation just means that you can do the same a couple of times, And any error that you may make can also be validated exactly the same way. So it doesn't impress me at all to use this expression, a validated assay. And whenever I get it as a reviewer, that people have been using a validated, quote, quote, validated assay, I ask them to remove that sentence because it's untrue in all cases. So the most important for any assay is really to study the reliability criteria. They are classically put up by the analyst Richterich, who published a book about this. And what you need to look at is sensitivity of the assay. And that means that you have to be aware of the lowest amount or concentration that you can measure, and also the least difference that you can measure significantly. These are not necessarily the same. And as I said, glucagon is circulating in very low concentrations, so you need to have a good sensitivity. You cannot have a good sensitivity unless you have a good precision, and therefore you always need to indicate what your intra-assay precision is, and also your inter-assay variation, and you express that as the coefficient of variation. Now, specificity is the next criterion, and here we are specifically talking about related hormones and molecular heterogeneity. And finally, the most important of them, accuracy, which really could be worded like this: the degree of congruence between what you think you measure and what you actually measure. And that is, of course, very, very difficult. Let's have a look at some of these things. So, sensitivity for glucagon assays really should be down to somewhere at or below 1 picomole per liter, because that's where you will end up in physiology and pathophysiology. So it must have a sensitivity like that. I'm sad to say that most assays do not have this sensitivity. The precision must be fairly good, and fairly good values are intra-assay variation below 5%, and an inter-assay below 15. And you note that this is almost always a lot larger than the intra-assay. This means that you have to be very careful if you have a series of samples and measure it in two different assays, because the variation may be 3 times higher all of a sudden. A classical source of error. And then comes the specificity question, and here we are really talking about the related peptides, as I showed you at the beginning, and other proglucagon products. And finally, some unspecific interference, which has marred many radioimmunoassays and other assays, and which is certainly also relevant for glucagon. How do you study the accuracy? Well, you can do that by applying chromatography or LC-MS, but you can also do it by looking at the results you get from stimulation. For instance, if you infuse arginine in healthy subjects, you must see a very, very robust response. And your glucagon levels must respond to changes in plasma glucose in the expected way. Otherwise, your assay doesn't work. Actually, I propose the use of measuring the metabolic clearance rate during an infusion of hormone as a test for assay accuracy. I know that this is fairly wild, but it's actually a good method. The one who gets the lowest metabolic clearance rate is the one who has the right value. So, specificity. Unspecific interference, as I said, plasma protein very often interfere with the assay. The assay matrix very often interferes. It's difficult to deal with, and that's why many people have been forced to say that, we'll have this afterwards. I just got a question on degradation post-sampling. Do I recommend addition of protease inhibitors? No, I don't. So, specificity, unspecific interference, which is very important. And how do you do it? Then you have to resort to something like plasma extraction. This can be done in various ways: ethanol, ZPAC, affinity extraction. The affinity extraction is something that has been studied over the years, and it entails a number of problems, because the recovery after affinity extraction is very essential, and particularly if you're looking at molecular patterns, they may change. So, specificity. So again, let me point out that these molecules are members of this large superfamily, and you really have to take into consideration whether your antibodies will cross-react with any of these. This is almost never studied. And then the even more important thing. So, measuring glucagon, well, we will use antibody methods, of course. So what kind of antibodies do you have? So if you just immunize with the molecule glucagon, you'll almost invariably get antibodies against this sequence, a mid part of the molecule. So using that kind of antibodies, you will of course be able to measure glucagon, yes, but you will also be able to measure proglucagon, and you will be able to measure glicentin and oxyntomodulin. In other words, you get a kind of total glucagon value, which is very difficult to interpret. And then, of course, if you use an N-terminal antibody like this one, you will find glucagon, yes, but you will also measure oxyntomodulin. So that cannot really be used. What about this one, the C-terminal antibody? It will measure glucagon, yes. Nothing here, nothing here. So that is what we usually call the glucagon-specific antibodies. Clearly, if you have molecules that are extended in this direction, these will also be measured. Glicentin is not, of course, because it's extended at the C-terminus. So it's only in the case that you have extended molecules in this direction that you will have a specificity problem. And they are quite specific, these antibodies. There is a molecule called Glucagon 1-61, which will be co-determined here. And it was also called large glucagon immunoreactivity many years ago. So there are molecules like that out there, and you really should look at it in your particular particularly clinical investigations before you use them. So, as I said, radioimmunoassays with mid-region antibodies will measure all glucagon-containing forms, glicentin, oxyntomodulin, proglucagon 1-61, and proglucagon itself. That we could call total glucagon. It's not that interesting. So total glucagon minus the C-terminal glucagon will give you the remaining forms up here. And that is what we usually call enteroglucagon or glicentin or plus oxyntomodulin levels. The C-terminal antibodies will measure glucagon and glucagon 1-61. The N-terminal antibodies will measure glucagon plus oxyntomodulin. Sensitivity? Here's an example. So here we were using two assays that are both C-terminal and looking at some experiments where we were infusing GLP-1, it was in this case, and the GLP-1 receptor antagonist Incretin into people with type 1 diabetes without beta cell function, with beta cell function, and in controls. Now, the point of this study was to look at the effects really of GLP-1, and you can see here that there's a strong inhibition of glucagon secretion, almost going down to 1 picomole per liter. In these patients, also strong inhibition here, also strong inhibition here. This was with one assay. Now let's look at another assay. It really measured more or less the same thing, but it was very difficult to see any inhibition of glucagon secretion in these patients, and in these, and in these. And the problem is that all of this took place at low glucagon levels, where the sensitivity was not good enough. So here you really have an important issue. Your assay must be very sensitive. So it should really look like this. So these are experiments, and this could be used as a kind of accuracy test for the glucagon assay. How does glucagon behave if you have high glucose levels in healthy individuals, if you maintain fasting glucose levels, or if you let them drop down? In this case, it was done with insulin. And you can see that hypoglycaemia should result in a robust response like this. If you do nothing, glucagon should stay. And if you infuse glucose, glucagon should drop. And again, look how close we get to levels of 1 to 2 picomoles per liter, very low levels. Here we actually did a comparison of two assays that are both C-terminal radioimmunoassays. This is the most conventionally used. Originally it was called Linco, now it's Millipore. And these are, as you can see, people with normal glucose tolerance and type 2 diabetes mellitus, and some that it was an experimental setting where people were, exposed to what we call glucotoxicity that could have changed something. But in all of these cases, these were the basal levels, these were the glucose-stimulated levels, these were with GLP-1 infusion, this was with arginine. And you can see this pattern, the millipoint RIA and the pattern with our own radium immunoassay, and you can see that they're completely identical, really. But you can also see that one of the assays appears to have a matrix problem because all of these values are considerably higher than the values measured over here. Of course, you can discuss which assay is the right one. That's a very technical and long discussion, but it does illustrate that one assay may have a matrix problem, whereas the other does not have it. Fortunately, to solve all these problems, you really need to have a sandwich assay. And that is because with a sandwich assay, you can have antibodies that are sitting in the C-terminus and in the N-terminus like this. And using this combination of a C-terminal and an N-terminal, you can really get rid of cross-reactions with this form and with this form, because they're extended, of course, and also with this 1 to 61 form that I mentioned, and any other form, really, that has an extension or a shortening of the N-terminus or the C-terminus, for that matter. So ideally, people should really be using a sandwich assay. And here, fortunately, there is one now on the market. It was developed by Mercodia, and it seems to behave very well. There are a couple of other ones out there also. They are not quite as good. It's, again, I've given you the list of all the reliability criteria, so why don't you look for yourself which one functions better? As I said, the glucagon assay will then use these N-terminal and C-terminal antibodies. Fortunately, using this antibody, you can also start looking at oxyntomodulin molecules because it will also measure the oxyntomodulin moiety. And then if you also have antibodies against the other regions of the proglucagon molecule, you may end up being able to measure all of the molecular forms, including the glicentin form. Just a little bit about glicentin. So glicentin, this is this molecule, and how should you measure that? Well, if you measure this part of the molecule, you will also measure oxyntomodulin. If you measure this part of the molecule, you'll also measure glucagon and oxyntomodulin. If you measure this part of the molecule, you'll also measure GRPP and proglucagon. So that's a bit of a problem. How do you do that? And what about glicentin? It's produced in the gut mucosa. The concentration is around 100 to 200 picomoles per gram, so there's a lot of it. It probably does not have any glucagon activity. It may be metabolized to oxyntomodulin and glucagon under certain circumstances. This was suggested some years ago. I'm not sure it's true. It's something that should be looked at in more detail, but it's a possibility. It doesn't seem to have any effect on glucose and insulin in man. We've looked at this. We've never published it, but because nothing happened. But I guess this is the way it is. It was reported to inhibit acid secretion in rats, and this may be true. It probably had a rather long half-life of some 30 minutes at least. Here I've put up that it might have growth effects, and that is because there was a little bit of a tendency to an increased intestinal growth after glicentin administration in certain animal studies. And this is something that really, it was up for discussion many years ago, but was forgotten again because then we had GLP-2 that has a lot of growth effects on the gut. But maybe glicentin also has it. So there's a lot we don't know. There's a lot to be done. Regarding the physiology, particularly the secretion of glicentin, there's really a large body of literature out there, but it deals with enteroglucagon, GLI, glucagon-like immunoreactivity, or gut glucagon, as it was also called, particularly before glicentin was really identified as a molecule. Those measurements always include glicentin and oxyntomodulin, which it makes it a little bit more of a problem, but I'll come back to that in a minute. So regarding the measurement, so far there's no specific assay. It is usually measured as total glucagon minus the pancreatic glucagon component. That is a side-viewing antibody minus that obtained with a C-terminal antibody. The assays against GRPP, which was this glicentin-related pancreatic peptide, will measure both glicentin, GRPP, and proglucagon. Specific assays, therefore, require sandwich technology. So we cannot go there before we have these. these are some measurements of total glucagon and subtracted, pancreatic glucagon subtracted values, that is intestinal glucagon, in patients that were operated with a gastric bypass operation. And this is then before the operation, and this is a few days after, and this is 2 months after, and this is 1 year after. And you can see that they have a tremendous secretion of this total glucagon molecules. And you can see that most of it is really intestinal, because if you subtract the glucagon part, which is strange enough already, shows this rise that I talked about a minute ago, if you subtract that, you still get very high levels, indicating that there's a lot of secretion of at least the combination of glicentin and oxyntomodulin. So what can we say about oxyntomodulin? Okay, here we have it. So if you measure with side-viewing glucagon antibodies, you will pick it up, but you will also get glicentin and glucagon. If you use this, you will also get glicentin. If you use this and this N-terminus, then you will also get glucagon. So what do? There's only one way of doing it, that is looking at a sandwich analysis. And what can we say? So far, people have been trying to measure it with cross-reacting antibodies, and that doesn't really work. There's no specific assay. There's a need for a sandwich ELISA. There are a couple of published and commercially available assays out there for oxyntomodulin. At least one of them was 1,000-fold wrong in its levels, and the other one didn't measure oxyntomodulin at all. We've published that recently. Quite dramatic. So what is the reality here? So what we did was really tedious experiments. What we did was to do gel filtration, molecular sieve, profiles of human plasma. So this is what it looks like with a side-viewing antibody measuring total glucagon. And here you have glicentin. This peak is glicentin, and this peak is oxyntomodulin. So here you have the relationship between the two. Typically, about 1/3 is oxyntomodulin, 2/3 is glicentin. And if you look at this total concentration, it means that oxyntomodulin concentrations are low. That's the way it has to be. So an assay for oxyntomodulin must also be very specific. Here is some very interesting data where we've been looking at total GLP-1 this time, after gastric bypass in a patient, and where we've also looked at oxyntomodulin using a prototype of a sandwich ELISA, and also a sandwich ELISA for glicentin. And these are really the first data ever, I think, reliable data measured directly from plasma in gastric bypass patients of these hormone levels. So, as I said, these antibodies enable us to measure specifically glucagon and oxyntomodulin by using antibodies out here, and also glicentin by using antibodies that really can create these sandwich ELISAs. Just like that, and that's where you have these antibodies. So, I think this was it, and I thank you for listening to this long talk, I'm afraid, where I have, tried to summarize almost all I know about measuring glucagon and glicentin and oxyntomodulin. Thank you.
Wonderful. Thank you for that very interesting and informative talk. So we did receive one question I believe you've already addressed, but just to reiterate that, the question was, does that mean, you said degradation post-sampling was not an issue. Does that mean you do not recommend adding any protease inhibitor to the blood sample immediately upon collection? Is this step not necessary?
So perhaps I should be a little bit more careful. Because we've just published a paper about this, and what we found was that using these conventional methods for measurement, we really couldn't detect any effect of the usual protein addition. I suppose this also applies to other proteinase inhibitor cocktails that you can use, because we really found completely identical values under different conditions of storage and addition of all sorts of inhibitors. So that was not obvious. But I have to say one thing. First of all, we were using EDTA plasma, and that is a good thing. EDTA will block some of the reactions of some of the enzymes that can cause problems, and gives you a very nice preparation of plasma also. So you get rid of a lot of problems if you use EDTA plasma and sample in the cold. So with ice, it seems that you can prevent most of the problems. So we did that. We've also looked at different temperatures in other studies, and that will come out soon, I believe. So it seems to be less of a problem than we thought previously, and at least the protein does not seem to be very important. But I'll have to admit that we have to do everything again with the new assay from Mercodia, because we've really not looked at possible N-terminal modifications and whether you can rescue those with additional proteins. So this is something we have to look at.
Great. We have another question, and that is, what do you consider normal, quote unquote, and out of normal ranges of glucagon?
So that's a very, very difficult question, and it's a question that we often pose ourselves because we get samples from clinicians that suspect endocrine tumors. So fortunately, in all the glucagonoma patients we have looked at, the values have been fantastic. They've been thousands of picomoles, or at least hundreds of picomoles. So you're not in doubt at all when you get some of these samples. The question is, what should you call it if a diabetic individual comes up with a fasting level of 12 picomoles per liter of glucagon, or 15 for that matter? Is that abnormal or not? And it's definitely abnormal for the glucose concentration that is present at the same time. So this is really not a question that can be answered very well, and I would always recommend that in that kind of cases that you look at a control group to look at it. Of course, you can use the classical way of collecting a large material of so-called healthy people and measure their values and have a standard deviation calculator and look at a given sample. But it really doesn't help you, because you need to know so much about this person. You need to know exactly what the glucose was, whether he was fasting. So it is very difficult, and I don't think it's really relevant to do this. So you must look at a control group at the same time, and preferably you should have some. With glucagon, it is interesting to look at the fasting values, definitely, but it's always also interesting to look at some function tests. And here, of course, the two interesting function tests will be effect of hypoglycaemia, intravenous glucose, for instance, or arginine, which is a good function test for glucagon.
Great. We have another question from an attendee, and this person writes, do you have any comments on GLP-1 inhibition on glucagon receptors? Are you aware of any tachyphylaxis process that could potentially occur with glucagon inhibition?
So the most important here is that GLP-1 inhibits glucagon secretion. It does that very robustly. I don't know of any interaction of GLP with the glucagon receptor. The opposite, though, is true. Glucagon may interfere with the GLP-1 receptor. It has only about 1/100th of the potency of GLP-1 itself. But there's an interesting interaction perhaps going on in the pancreas, where glucagon from the A cells may actually influence beta cells via the GLP-1 receptor. It's a little bit theoretical, but it's absolutely possible. So we don't think that there is any tachyphylaxis of the effect of GLP-1 on glucagon inhibition. And we think that it is an essential mechanism of action of all the GLP-1 agonists that they inhibit glucagon secretion. But coming to think of it, I'm not sure that I've seen a formal study that had looked in detail at this. What I do know is that the effect on glucose regulation does not show any tachyphylaxis. What happens really with time is more the result of beta cell deterioration in diabetics. That is, that beta cell function decays further, and that therefore it may seem that GLP-1 will lose some of its effect and show tachyphylaxis, but that's not really the explanation. The explanation is a progressive beta cell failure.
Okay, and then we have someone who would like for you to comment on the stability of glucagon in stored samples.
Yeah, it's a good question. Again, we've just completed some very tedious studies where we've looked at storage for up to 1 year. And again, we need to do all of it again with the new sandwich ELISAs. But, using the C-terminal assays, we couldn't really detect any decrease in the concentration of stored samples, both clinical samples and samples that were spiked with exogenous glucagon. So it doesn't seem to be that terribly unstable.
Another question is, are there any cell-based assays?
There has been attempts to use cell-based assays, and of course, the cells you would have to look for here would be some beta cell preparation cell line or a hepatocyte type of cell, and that has been looked at, but I don't know of anybody who seriously uses this for clinical use. I worked a lot myself with receptor assays for glucagon, and then I used for that purpose hepatocyte membranes, and it worked quite well, but it was rather laborious, and it didn't really add any new information to anything we did, so I gave that up again.
Now, another attendee has asked, is glucagon conserved among species, or are assays species-specific?
So among mammals, glucagon is well preserved, except for the guinea pig, as far as I remember, which has a 1 amino acid change. I think I remember correctly. But there are important changes in birds, and if we go further away from mammals. But in mammals, there isn't a lot of species differences.
Just a couple more questions. What matrices are appropriate for glucagon measurement? Serum, plasma, others, are there differences?
Yeah. So again, this is a very important question, of course. So first of all, as I said, we think that one of the reasons that we haven't really observed any major issues with degradation or storage problems is that we always treat our samples in the cold. So as long as you do this, then you're safe, it seems. Now, with serum, you cannot really do that very well. So then from that follows that you must use plasma. And as I said, it's clearly our experience that EDTA plasma is superior. So this is the explanation for why we need to use plasma. And then regarding other matrix problems, I'll have to admit that we extract all our samples, and that is because we have experienced that if you don't extract, you can find the values all over the place. There are from almost all glucagon antibodies will show some unspecific plasma protein interference, and there may also be other problems. So to get rid of those, we usually extract our samples. We don't concentrate them because that usually creates more problems than it helps. I'm sure that other ways of getting around these problems can be made, and we all hope that the sandwich ELISAs will not suffer from these problems. Unfortunately, I know that the sandwich ELISA for GLP-1, for the active GLP-1, does show some spuriously high results sometimes in some plasma. So perhaps you're not completely safe. But at least from a theoretical point of view, it's a much better approach to use a sandwich ELISA to get rid of these problems.
Another attendee has asked, do you recommend addition of any specific protease inhibitors other than a protein to samples to be analyzed at a later date?
Yeah, but again, as I said before, I really don't think we have a major problem unless one will turn up now that we are using the sandwich ELISA. So I don't see the need immediately for adding other protease inhibitors. As you know, there are many that you can buy. There are commercial ones with combining a lot of inhibitors. And this is absolutely worthwhile looking at. But from the experience we have so far, it doesn't really seem to be needed.
Okay. And our final question, do you have any explanation for the increase in glicentin after the Roux-en-Y gastric bypass?
So that immediately, this is L cell secretion from the gut. And we know that the L cells are tremendously stimulated by this. And it also shows up as a tremendously increased secretion of GLP-1 and GLP-2, actually. So we know that the cell is stimulated greatly, and the same is actually true for PYY, for peptide PYY. So the very good question is, why is that? And one thing that happens is that you have a very accelerated gastric emptying, or that's not really an emptying, there's a very accelerated entry of nutrients into a segment of the small intestine with a very much higher L cell density than there is in the proximal gut. So this is one of the conditions. We also know that, for instance, glucose is absorbed very, very rapidly after the enters into the small intestine in these patients. And we know that L cells respond to glucose by an extremely elevated secretion of GLP-1. So this probably is one of the most important mechanisms for this. But of course, it raises a lot of questions as to what is the total picture like? Is it bile acids, and this is being investigated these days? Are there lipids that specifically stimulate the L cell? What about the receptors that they're equipped with? GPR 119, GPR 40. People are working with agonists against these receptors to create a similar rise in GLP-1 secretion in particular. So there are a lot of questions that are being investigated these days. But the rapid entry and absorption of nutrients seems to be the most important right now.
Okay, so I'd like to actually go back to one of the questions about extraction and addition of a protein. And my colleague Maria Schwanbeck can actually address what we have seen in-house with our sandwich ELISA.
Please go on.
Yes, so we made in-house experiments with extraction of the EDTA plasma samples with acetonitrile, and we saw no differences in measured glucagon values in extracted samples or the EDTA plasma samples.
Very nice.
Okay. I think we
have also done in-house studies with addition of a protein to EDTA plasma, and we saw no differences in the glucagon levels.
That's nice. That's very reassuring. Thank you very much for this.
Okay, so I think we may have time for just one more question, and that is, are there any post-translational modifications within the peptide that may interfere with an assay?
So the only post-translation modification that, of course, we have the processing that I have discussed already. I mean, the cutting up in smaller pieces. But is an O-glycosylation which occurred in the major Proglucagon fragment. That's the only one I know of. And that is not terribly relevant for the measurements that we are doing currently, and particularly not for glucagon because it's simply not there. It's in the major Proglucagon fragment. So this is I certainly won't exclude this, but there's absolutely nothing known about it.
Okay. Well, with that, we will conclude this webinar, and I'd like to thank everyone for joining us today, and a very special thank you to Dr. Holst for a wonderful presentation and Q&A session.
Thank you very much.